It's wonderful to have this opportunity to share with you some of the truly outstanding research that is going on in the College of Science at Northeastern University. You know, if I asked any of you what you thought a key medical problem is, in human health, what is a key medical problem? After you answered me that you thought it was overcoming COVID, you probably would think about, okay, what are the other things I'm thinking about? And I suspect that one of the things that many of you have encountered either personally or through a family member or someone you know, or certainly you've seen in the press, is the notion that some of the most important therapeutics we have, the antibiotics, are becoming ineffective against their target bacteria. And this is very serious because if you cannot overcome a bacterial infection with the help of antibiotics that really changed the course of human history, if you can't use those therapeutics, then human health and the health of agricultural animals, for example, suffers enormously. It is an enormous problem that really has been and will continue to be devastating. So a key aspect of medical science, of biological science, is to try and understand how these powerful antibiotics that target bacterial infections, how they work, how they are becoming ineffective because of resistance by the pathogens to these therapeutics, and what you can do to move to a next generation of medicines that are truly going to overcome the bacterial infection problem for the future. We are really fortunate to have at Northeastern a cadre of outstanding investigators who are spending their research time thinking about how to overcome antimicrobial, antibacterial resistance and develop or discover the new generation of antimicrobials. And it's my honor today to introduce two of them to you, members of the biology department here at Northeastern, who are really extraordinary investigators and who are also at two really different points in their careers, which is very exciting as well. So let me start with Eddie Geisinger, who is an assistant professor of biology at Northeastern University. He has an MD, PhD, 2 degrees from New York University, and he has a very eminent career that before he came to join us, he already started to become fascinated with a kind of what he calls a superbug, Acinetobacter, which is something that can become antimicrobial resistant. And the Geisinger group has been aiming to understand and combat Acinetobacter, which is a very problematic bacterial infection that can impact human health. And he is deeply interested in trying to develop novel therapeutics by understanding the mechanisms by which Acinetobacter becomes antimicrobial resistant. It's our pleasure and honor to have Professor Geisinger on our faculty. Professor Kim Lewis is really one of the giants in the field of antimicrobials and microbial resistance. He is a university distinguished professor, and he's director of the Antimicrobial Discovery Center here at Northeastern University. He is the recipient of honor after honor. Just yesterday, I emailed him to congratulate him on 2 of his latest honors, a wonderful, including being probably for multiple times, one of the most cited investigators of the previous year across the entire country. So we are truly honored to have Kim Lewis as a member of our faculty here. He has been looking at antibiotic tolerance, or that is the resistance to antimicrobials. And his group is developing compounds that overcome antimicrobial resistance. He is focusing on a set of gram negative, Bacteria, yeah, I'm looking at him and he's nodding to me. He's the gram-negative bacteria, the ones that are really difficult, and I'm sure he may allude to why, that are the ones that really are impacting human health. So Professor Lewis has done extraordinary work and has made discoveries in terms of whole classes of new antimicrobials that can be helpful to human health. I give you this fairly long introduction because I want to celebrate these two faculty members, and because I know this is something that you have already thought about and are excited to hear the insights that are coming from Northeastern University and the insights that are carrying the university, the College of Science forward in promoting the health of people and society. I'm so grateful to all of you for being here. I am so grateful to all of you who support us in our mission of promoting excellence in science, in promoting the very best outcomes to the great challenges, the great research challenges of our time. So thank you for being here, and thank you for being part of the College of Science community. And without further ado, let me turn the discussion over to Eddie and Kim. And I'm not sure which way, who's talking when. So Eddie, I see you've unmuted, and I'm going to bet that you're going to start. So thank you very much.
All right. Thank you, Hazel. Let's pull this up here. Yeah, thank you so much, Hazel, for that awesome introduction. It's great to be here with all of you, and I'm really excited to tell you about some of the research from my lab here at Northeastern, which is addressing this major problem of antibiotic resistance. So what I'll do is I'll begin with a big picture introduction to the problem of antibiotic resistance, and this is going to expand on many of the points that Hazel just raised. And then I'll tell you a little bit about the research in my lab, which is addressing this problem. And as Hazel mentioned, I'll tell you about our particular focus with this really critical superbug, which is one of the most drug-resistant bacteria that is out there right now, which is called Acinetobacter baumannii. And then I'll hand it over to Kim. Okay, so what is the antibiotic resistance crisis, right? And why is this problem so significant, right? So it's such a huge deal because as Hazel nicely mentioned, antibiotics are these miracle drugs, right? They're one of the most powerful tools that we have that enables us to protect public health, right? They allow us to cure a whole range of life-threatening infections. But as Hazel mentioned, really what they're enabling us to do is to practice modern medicine, right, as we know it today, right? So many interventions like surgeries, organ transplantation, chemotherapy for cancer patients, care of preterm babies, right? All of these are made possible because of antibiotics, right? And if antibiotics lose their efficacy, all of these procedures that are so important in modern medicine become extremely challenging and problematic, right? But unfortunately, that's what we're seeing happening now, right? Because of the ability of microorganisms, right? This includes bacteria and fungi, to rapidly and naturally evolve resistance to these drugs that are designed to kill them, as well as due to the overuse and misuse of antibiotics, both in the clinic as well as in other domains like animal agriculture. We've seen that these microbes are have now evolved the ability to resist these antibiotic therapies, right? We see widespread antibiotic resistance. This is a huge crisis right now, right? And so what does the antibiotic resistance crisis look like? So we know that it's taking a major toll in terms of morbidity and mortality. So the CDC a couple of years back studied this and showed that there are about more than almost 2.8 or more than 2.8 million infections with antibiotic resistant microbes each year. And these result in over almost 36,000 deaths annually. And in addition, there are another more than 200,000 infections and almost 13,000 deaths due to this additional microbe known as Clostridium difficile. This is also known as C. diff. So this bacterium is not antibiotic resistance per se in its own right, but these kinds of infections emerge in hospitals because of use of antibiotics in hospitals and because of widespread antibiotic resistance. So we see that this is really taking a huge toll, and this burden is amplified worldwide. Right. Currently, we see that there's more than 700,000 deaths per year due to antibiotic-resistant infections. And there's been a group in the UK that examined this problem recently and estimated that if the current antibiotic resistance rates continue and we don't do anything to solve this problem, by the year 2050, this number of deaths per year globally is going to skyrocket to over 10 million deaths per year. Right, this is more than the current annual death rate due to cancer. Right, so we know that this is a huge problem currently, and it's only getting worse. And unfortunately, we're seeing it also get much worse in this era of the COVID-19 pandemic. And there are a number of factors for this that we could discuss. So, given the scale of this antibiotic resistance problem, one would think that this would be a major priority of the pharmaceutical industry to counter this crisis and to develop and introduce new kinds of antibiotics to the clinic. But unfortunately, this is not what we're seeing. We've seen that there has been no new class of antibiotics that has been discovered and introduced to the clinic since 1987. Right, so there are a number of factors that are responsible for this. Some of them are economic, and another major factor is that our current antibiotic discovery platform, the conventional antibiotic discovery platforms are associated with major limitations. And Kim's research addresses many of these limitations and has identified a number of really exciting new classes of antibiotics that are really promising. But the major take home from this timeline here is that there's this critical unmet need, right, for antibiotic resistance. The current antibiotic development pipeline is in nowhere near keeping up with the current problem of antibiotic resistance that we have today. So one really exciting solution that many scientists are pursuing, including research in my lab, as well as several tracks in Kim's Antibiotic Discovery Center, is to develop antibiotics that are more selective in nature, right? These are antibiotics that specifically target the pathogen that's responsible for an infection, right? Pathogens are the microbes that are actually responsible for causing an infection and a disease, right? So almost all of the antibiotics that are in our current clinical toolkit and all the many of the antibiotics that I showed you in that previous timeline are broad spectrum by nature, right? These antibiotics target many different species of bacteria non-selectively, right? And while these types of therapies have their advantages for sure, there are many drawbacks to this kind of broad spectrum therapy, right? So a really important drawback is that these kinds of therapies will damage our microbiome, right? So our microbiome is this community of good bacteria that lives with us, primarily in our gut, but at many sites throughout our body. And these good beneficial bacteria provide a range of important functions for our health, right? They enhance nutrition, they represent important elements of our immune system, they prevent the development of numerous diseases, right? So if you're out there sipping your kombucha or eating kimchi right now with your lunch, you probably already are aware of the microbiome and understand its importance, right? This kind of broad spectrum antibiotic therapy is actually associated with not just killing the targeted pathogen responsible for infection, but it's going to wipe out your good bacteria in your microbiome. And that's a very bad thing that's associated with a range of negative health outcomes. So in addition, these kinds of broad spectrum antibiotic therapies actually contribute to the problem of antibiotic resistance. They promote antibiotic resistance, right? By killing the infecting bacteria as well as other bacteria that are on us and in the environment, this is going to create the opportunity for antibiotic resistance to amplify and to spread, right? So this kind of therapy is actually contributing to the crisis. So by contrast, narrow spectrum or pathogen selective antibiotics would have the benefit of avoiding this kind of collateral damage to the microbiome, and it would minimize the emergence of antibiotic resistance, right? And so this is the kind of next generation types of therapies that that we really want to develop to add to our toolkit against these resistant microbes. So this brings me to the research that I'm focusing in my lab here at Northeastern. And so the type of selective antibiotic approach that we're interested in developing is to be targeted against one of these most critical superbugs out there, right? One of the most drug-resistant, dangerous pathogens, which, as I mentioned before, is called Acinetobacter baumannii. And so Acinetobacter baumannii is a bacterium that tops the list of everybody's priority list of microbes that we really need to target, right? So the WHO came out with their high priority list of microbes for which we need new research and development for new antibiotics, and Acinetobacter topped their priority list. The same goes for the CDC, which gave Acinetobacter their highest code level red label as this urgent threat to public health. And this is a pathogen that I became interested in several years ago during my medical training. So during my rotations on critical care wards, I saw a number of cases of infections with this microbe. And so early on, I came to really appreciate how much of a problem these infections posed. And so we know that these microbes are responsible for a large proportion of opportunistic diseases in hospitals, right? And so these pathogens target the most vulnerable patients in the hospital. So these are patients that are critically ill, that are in the ICU, hooked up to multiple indwelling devices. These are the patients that are at risk for developing these kinds of infections. And in terms of how these infections manifest, most commonly we see ventilator-associated pneumonia, bloodstream infection associated with catheters, wound infections, and all of these kinds of infections can progress into this really dangerous picture known as sepsis, right? And this kind of systemic, dangerous situation is associated with a really high risk of mortality in these patients. And so in addition to infections in the hospital, Acinetobacter has been in the news over the years, and it's been given the nickname Iraqibacter, because we've seen that it's actually responsible for causing a large number of severe cases of wound infections and invasive blood and bone infections in soldiers from war zones in Iraq and Afghanistan. Right, so this is really showing that the impact of these bacteria is quite broad. And so we see that these bacteria have rapidly evolved resistance to multiple classes of antibiotics. And what's particularly alarming is that we're seeing pan-resistant isolates of Acinetobacter emerge in the clinic now, right? So these are strains of these bacteria that are resistant to every single antibiotic that is available clinically, right? And so this is really raising the specter that if we don't do something to stop these infections, that one day we might be seeing truly a post-antibiotic era where we see these widespread untreatable infections, right? So this is the problem that the research in my lab is seeking to address, right? And a major goal of our research is to discover and develop targets for new kinds of selective antibiotic therapy against these superbugs, right? And the idea here is we want to better understand these pathogens and identify proteins and activities that are specifically found only in Acinetobacter and closely related organisms, right? And so by these kinds of proteins and activities could present potentially targets for selective therapies that specifically attack these pathogens. And then armed with this kind of knowledge about these targets, we could then screen for and design chemical inhibitors that can now allow us to attack these pathogens selectively. And so this kind of research addresses a number of problems in the field with studying these microbes. And one major problem that this addresses is that Acinetobacter has diverged evolutionarily from other bacteria. Right, it's quite different from other bacteria, even others in its sort of family of gram-negative bacteria that it's relatively related to. And so what this means is that many of the paradigms that we've learned by studying model bacteria, like E. coli, for example, these paradigms about drug resistance, about basic elements of cell biology, these don't necessarily apply to Acinetobacter, right? And so this evolutionary divergence manifests in a number of ways. So first, we see that there are many proteins encoded in the genomes of these bacteria that have no known or predicted function because they're so different. And so we call these proteins dark matter proteins. But many of these are found across all strains of Acinetobacter. So we predict that they're doing something important for the cell. In addition, these bacteria lack many key proteins that are essential for many pathways and processes in other well-studied bacteria. So for example, these missing proteins belong to a number of function in a number of different pathways usually. But one pathway that we've seen a lot of missing proteins is cell division, right? So this is just the basic ability of a bacterium to replicate and go from one bacterium to two and so on. This fundamental process in Acinetobacter is lacking many of the typical proteins that are so essential in other bacteria. So we predict that this pathway of cell division might be a way that a process that if we better understand it, we could target it with selective antimicrobials. And we hypothesize that some of these dark matter proteins will be providing functions that substitute for the missing proteins and provide this unique strategy for cell division and other pathways in the bacterium. So in order to study this problem, identify the best dark matter proteins that might be candidates for selective targets and understand their function, we use an approach called phenotypic signature analysis. And what this approach allows us to do is it allows us to study knockouts of every single gene in the genomes of these bacteria and analyze them all together using the latest DNA sequencing technology so that we can understand the effects of every single gene and protein in the cell for a variety of conditions, understand which ones are most important for growth, for drug resistance. And by doing this kind of analysis, we can also identify leads on the function of these of every protein in the cell. And so I'll show you an exciting result from this kind of analysis, which is we found a number of dark matter proteins that we look nothing like any other protein out there, but play really important roles in a number of areas for these bacteria. And that could represent potentially select targets for selective antibiotics. So one exciting result was this protein that we've called ADVA. This protein looks like nothing out there in the database, but in Acinetobacter, it's critical for cell division, right? And so we've studied this by analyzing where the protein localizes when we fluorescently label the protein. And we see that this protein localizes at mid-cell at sites of cell division. And using a number of genetic engineering techniques, we see that when we deplete this protein, the cell dies completely. But on its way to dying, it shows this dramatic elongation effect, which is consistent with having this dramatic defect in the ability to divide, right? These bacteria are sort of confused. They're growing by elongation right before they die. So this is showing that we are able to identify these exciting dark matter proteins, identify what their functions are, and now have leads to begin to develop potential selective inhibitors and understand how these microbes are undergoing these basic processes in a unique way. So with that, I'll pass it along to Kim Lewis, and I'm happy to take questions and discuss things after his presentation.
Eddie, you'll forward that for me? Yeah, terrific. Thank you. All right, I will show you a couple of slides of what we've been doing over the past decade at the Antimicrobial Discovery Center. And here is an overview of compounds that we identified that are currently in development. So each one of them addresses an important and unmet need. So this one, ADEP, is a very interesting compound that we identified as the first antibiotic that can sterilize a bacterial infection. Currently available antibiotics in the clinic, they will kill the majority of cells, but then there's a small subpopulation, we call it persisters, that are dormant spore-like cells. And those are not killed by regular antibiotics, and that's why you have chronic infections, relapsing infections, etc. ADEP kills all of them because it does a very interesting thing. It turns on proteolysis inside the bacterial cells, so it forces bacterial cells to self-digest. So moving on to the next compound, teixobactin. This we discovered in collaboration with Slava Epstein and a startup Novobiotic. And this comes from a previously uncultured bacteria. So we've figured out how to grow bacteria that don't like to grow in our labs. And this compound came from an interesting novel uncultured bacteria. And the remarkable property of this compound is that there is no detectable resistance to it. So this is the first antibiotic with no detectable resistance. It hits targets which are not proteins, they cannot mutate, and that, at least in part, explains why there's no resistance to this compound. Now, these two are terrific, but they act only against gram-positive bacteria, creatures like, strep and staph, which are important. But the current critical need, as Eddie mentioned, is against gram-negative bacteria like Acinetobacter and E. coli and such. So we recently refocused on our effort on gram-negative bacteria, and here are two compounds against gram-negatives. I'll skip this because I'll show you a separate slide on this one, on darobactin. But this is our latest, and that is hygromycin A, that selectively kills Borrelia burgdorferi, that causes Lyme disease, doesn't harm the microbiome. That's one of the things that Eddie highlighted as a requirement for new antibiotics. And this one is hopefully will become a new drug to more intelligently treat Lyme disease. Could you show the next, please? Okay, so in thinking about antibiotic discovery, we have a process. It starts with a gram of soil, and then we isolate bacteria from that gram of soil. We test them for the ability to produce antibiotics. We isolate the compound, determine the structure, test it for toxicity, test for efficacy in animals, then eventually it will go to humans. So it's a long process, and on almost every step of this process, there's a bottleneck. And we've spent a lot of time resolving these bottlenecks, and we resolved all bottlenecks, but this one. Here I named this the mother of all bottlenecks. So you take a gram of soil, and you assume that in this gram of soil, there is some bacterial species that is making a terrific antibiotic, and you, using clever approaches, are going to isolate. Well, that assumption is based on absolutely nothing. You don't know that this gram of soil contains what you're looking for. Well, you can take a couple of grams of soil, many such grams of soils, and still you do not know. And so if after working on this problem for, let's say, 10 years, you fail, you will never know where you failed. This is very frustrating. Since this problem does not have an obvious solution, it is never discussed in my field, right? So we are kind of collectively in denial. And so we decided to do something about it. And interestingly, this problem has a very straightforward theoretical solution. Could you show us next, please? So the theoretical solution is do not screen grams of soil, screen planet Earth. Somewhere on the entire Earth, there's going to be at least a couple of species of bacteria that make something interesting. The problem with this proposition, of course, is that this is completely impractical, that there is a difference of 24 orders of magnitude between what we can do and what we very obviously cannot do. But what I would like to tell you is that screening planet Earth for new antibiotics is probably something that has been done already and done many times. So what happens when organisms live on this planet is that they shed off their DNA. So we do that, bacteria do that, and then there's, let's say, a cloud of DNA floating around the planet. And then all organisms, including us, can pick up that DNA and insert it in their genomes. That's called horizontal transmission. And bacteria do that much better than we do. A typical group of bacteria would have been around for about 100 million years. So for 100 million years, they would be sampling this cloud of DNA floating around the planet, looking for, if you will, or screening for pieces of DNA that code for antibiotics that they are interested in. Next, please. So then how to collapse that enormous planetary screen into something that we can manage and handle? Well, how about we find a group of bacteria that has exactly the same requirements for antibiotics as we do? And that list of requirements is common sense, and it's very obvious. Those antibiotics have to be active against gram-negative pathogens, should have low toxicity to us, a good ability to travel through tissues, we call that good PK, and of course be efficacious in treating an infection. So when you draw up that list, then a particular group of bacteria comes to mind. It is this one, interestingly, how Eddie is reading my mind. This group of bacteria is Photorhabdus. They are gut symbionts of nematodes. So they live in the gut of nematodes. These entomopathogenic nematodes infect an insect larvae, release the bacteria, and the bacteria start making antimicrobial compounds to fend off competitors. Now, these competitors happen to be mostly gram-negative bacteria in that environment. The compounds they make should have low toxicity, not to harm the worm, which is a little animal. They have to be able to move through the tissues of the larvae, so have good PK. And of course, they have to have efficacy in vivo, which is in this larvae. Based on that logic, we put together a collection of these bacteria that nobody really cares very much about, apart from Amazon. Amazon will sell you entomopathogenic nematodes. You can then sprinkle them on your favorite geranium and provide it and protect it from infection by insects. And we actually do buy nematodes on Amazon periodically. So from this source, we isolated, you show next, please, Eddie. We isolated this compound, which we call darobactin, from the Greek word dar, which is gift. It's indeed a gift from these bacteria to us. This is the first class of compounds in quite a long time that acts against gram-negative bacteria, cures not only caterpillars, but also mice of infection by important pathogens. And so now this is in development. It has a fantastically interesting mode of action. So gram-negative bacteria are hard to hit because they have an additional outer membrane that restricts penetration of compounds into the cell. Darobactin solves that problem of penetration by not penetrating. It hits a target on the surface of the outer membrane. So that's Mother Nature is showing us how to solve tough problems. So that's all that I want to share with you. And I'm sure that Eddie and I will be happy to answer any questions.
Well, thank you so much, Eddie and Kim, Professors Geisinger and Lewis. We much appreciate your insight and your creative approaches to this really very tough problem. I want to encourage members who are looking, members of the audience who are thinking about this question, to put whatever you want into the chat as a question. Remember that there is no stupid question. There are only questions that you would like to have answered, and they are all really valid. So please don't be shy at all. Let us, I'm going to look down. There's some actually terrific questions here. Let me do a little bit of Zoom engineering here. That's better. Good. Here's a very interesting question for Dr. Geisinger. When you talk about resistance to all known antibiotics, are these pan-resistant bacteria also resistant to combinations of existing antibiotics?
Right. Yeah, that's a great question. So that has been looked at, and I think that is it becomes sort of a kitchen sink approach when you isolate these extensively resistant microorganisms, that you try to use multiple classes of antibiotics that can hopefully enhance each other's effects. And so possibly synergy is a potential way to get around this. But unfortunately, with these pan-resistant strains, usually this involves resistance to the polymyxin class of antibiotics. That's like the last line of treatment against these organisms. And often polymyxins are some of the best drug synergizers, because they help attack the outer membrane, like Kim mentioned, which can help address the impermeability barrier. But once these microbes become resistant to the polymyxin class, then you've lost the potential for synergy as well. And so, even synergy, while it's an exciting angle to attack these pathogens, many of the existing resistance mechanisms also block synergy.
Thank you very much. Here's an interesting question. Is there a way to make sure that bacteria don't develop resistance to a particular compound? I think I'm paraphrasing one of the questions in the chat. Professor Lewis, do you want to try tackle that one? Is there a kind of an ultimate antibiotic that bacteria just would never become resistant to, or is that some science fiction dream?
Well, no, I thought it was science fiction until we discovered teixobactin. So I, just like everybody else, was very much married to the paradigm that bacteria are always going to figure out how to develop resistance. But the beauty of science is that you change your opinion the moment you encounter a new fact, right? If only that were true for other fields of human endeavor. Anyway, so here we have an antibiotic to which we were not able to develop resistance. No biotic tried, my lab tried, and we figured out why we cannot develop resistance, because in order to develop resistance, there has to be, some mechanism of resistance. So one of the common mechanisms of resistance is the target that an antibiotic attacks. It's usually a protein, it mutates. Now we have a form of the protein that doesn't bind your antibiotic, you get resistance. Well, this compound does not attack proteins. It attacks a precursor of peptidoglycan, the cell wall polymer. It's not coded directly by genes. There's nothing to mutate. And the structure of the part that it binds to that compound is absolutely conserved among bacteria, right? So another very common mechanism is it works like a pump, literally. The antibiotic gets inside the cell and the protein pumps it out. And it's called efflux pump, right? Teixobactin acts on the surface of the cell. There's nothing to pump out. It's already outside. Right? And so when we published this paper, we called it the novel antibiotic with no detectable resistance. And we had to negotiate with the editor. The editor at Nature was telling me, I said, let's name this an antibiotic with no resistance. He said, no, we're not going to publish that. There's just no way. Let's say an antibiotic with low level of resistance. I said, no, I disagree. We cannot measure any resistance. So low level is misleading, it's just incorrect. He said, all right, how about no detectable? So we agreed upon no detectable. And a lot of people got very irritated with that and sent me an email, which said essentially this: send me your teixobactin and I'll mail you back a resistant mutant. Right? So I sent teixobactin around the globe. I've been doing that for a while. And I'm still waiting for that parcel to come back with a resistant mutant. Right, so a lot of people tried, of course, and so that's a good example how nature designed the compound with no resistance. I don't think there will be any resistance to teixobactin, and that is a very different paradigm and a possible way forward.
Very exciting. The power of biology. I mean, it's fantastic, isn't it? The power of life that you are mining, in a sense. I think it's fantastic. Dan Distel, I see your hand up. Let me go through some other questions if you want to pop something in the chat. Otherwise, I'll get to you when we have gone through a few more questions that are already in the chat. Here's one I think is really fascinating for Professor Geisinger. Once you've discovered in your scholarship the proteins in Acinetobacter that are necessary for survival, and you're trying to now take them into defining antimicrobials, what is the strategy there? Can you sort of interface with the approaches that Professor Lewis is taking to, bring together the biology that you're uncovering with the compounds, or do you go to designer compounds? Please give us a little bit of insight there.
Yeah, absolutely. So there's a couple of approaches that we're examining in parallel. We're looking at the structure of the proteins that we identify. By looking at the structure, this could potentially give us insight into what aspects, what kinds of areas of this molecule could be amenable to targeting, inhibiting. But really, we're also excited about looking for identifying chemical inhibitors of these proteins. And what we are excited about interfacing with Kim's approach. So the approach we really want to take is, because these are gram-negative bacteria that we're studying, we want to do an approach that identifies inhibitors that will kill the cell using whole cells at the same time as targeting this protein that we've discovered. And so what we do for this kind of approach is, and this addresses one of the questions in the chat from Blake, is we're going to be using two different strains in parallel. One of which is immune to being inhibited by an inhibitor of this target we would like to hit, and one that is completely killed, right? And we could do a few genetic tricks in order to have this kind of multiple strain parallel approach. And then this would allow us to identify inhibitors that are killing the cell as well as specifically inhibiting the target. And in terms of what would we be screening for, this is sort of aligns with a lot of what Kim's lab is doing right now in terms of looking for natural products that hit specifically bacteria. And so I think we can interface with them using a lot of their technologies to identify natural products that are inhibiting these bacteria. In addition, at the same time, we're really excited about new synthetic chemical libraries that are being developed that also take advantage of our knowledge of how we can get compounds across that gram-negative barrier. More and more is being understood about that. So once we can get really optimal libraries that we know can target gram-negatives, we can screen these kinds of libraries for inhibitors that specifically target what we've identified.
Thank you. That's extremely exciting. There's a lot on our minds about vaccines. We're very much, in favor of vaccines right now across planet Earth here. And there's an interesting question in the chat asking, what do you think of the possibility for immunological approaches to prevent resistant bugs? Are there enough similarities in targets to take something like a vaccine approach? If either of our scientists would like to tackle that one.
Eddie, you can take that one.
Yeah, I think that's a great approach. And these are not mutually exclusive approaches. I think it's a fantastic thing to think about vaccines in addition to having small molecule compounds that are ready to attack, right? If we could do these together, I think that's a fantastic multi-pronged approach. What's great with vaccines is that what you're trying to do is you're trying to teach the immune system to develop antibodies to target the surface of these microbes, right? Which is awesome because here you don't need to think about penetrating molecules across a membrane. You can develop ways to target the surface of these microbes. And so I think it's a fantastic way to have this parallel approach. In addition, for bacteria like Acinetobacter, which are so resistant and dangerous in hospitals, there definitely is a lot of interest in developing monoclonal antibodies, which is like a passive immune approach to bring antibodies to patients that need them at the time they're being infected, as a way to allow our immune system to enhance the killing of these bacteria. So I think it's terrific, and it's a parallel approach.
Very interesting. Daniel Distel, is your hand up still or is it down now? I see it vanished, but if you'd like to ask a question, please go ahead.
No, I think that my question was answered. So thank you.
Brilliant. Good. Good. I'm glad it was answered. Here's some other ones. There's a lot of questions in the chat, and then we'll keep those questions. And I do believe that we could prevail upon our eminent speakers to answer questions we don't get to subsequently. So we'll see what we can do there. But let's keep going a little bit. Here's one about the superbugs, which hide in ventilators and catheters and ultimately cause death by sepsis. Do you think that those are being counted by hospitals as death by COVID-19, or is there some kind of synergy here with bacterial resistance and with mortality associated with COVID? Anyone?
Eddie, you're the doctor here, so
Yeah.
that's up your alley.
Yeah, that's a tricky one. We are definitely seeing outbreaks of infections with these microbes, especially during the initial response to the pandemic, with all the increased hospitalization and increased patients in the ICU and potentially decrease in infection control. We've seen clusters of infections with these superbug microbes, and that definitely worsens the picture, right? These are already patients that are severely ill. They're suffering from COVID-19. They're now getting secondary bacterial infections. And so how that's counted, I don't know. But it's definitely a phenomenon that's happening, and they're exacerbating each other. And so you can really imagine that these microbes that linger and lurk in hospitals are becoming more of a threat with the viral pandemic.
One mess leading to another mess, isn't it? It's this whole landscape of, wow, what a world we are living in right now. Let's see, there's a couple of interesting questions. Kim, Professor Lewis, I'd like to ask this one to you. What innovations need to be made to get previously uncultured bacteria to grow? Or do you need to do that in order to do your global screening?
Right. So it's an excellent question. So for screening planet Earth, you need to figure out which group of bacteria screened for stuff we are interested in. And we were lucky to zero in on Photorhabdus. It is a cultivable group of bacteria, right? They do grow on our petri dishes. But we're on a hunt for additional ones. And of course, I'm sure that many of additional ones are going to be uncultured simply because of the numbers game. Uncultured bacteria represent 99% of the entire diversity of bacteria. So Slava Epstein and I developed methods to grow uncultured bacteria based on a very simple idea that they will grow under natural environment in the soil. So we take a sample of soil, put bacteria from that sample of soil in a dialysis basin, something enclosed with semi-permeable membranes. And now all the chemicals then diffuse across those membranes, and we trick bacteria. They don't know that something happened to them, right? And they grow inside that chamber. Once they grow inside that chamber, we domesticate them and they grow on our petri dishes. So that's one viable approach. Another approach that people have been using is to simply clone environmental DNA. You take the soil, you don't care where that DNA comes from. You simply put it in a plasmid, you plug the plasmid into something like E. coli and expect that E. coli to start making antibiotics. That is a terrific idea. It hasn't worked yet because the throughput wasn't there. But now my lab developed an ultra high throughput platform for screening it, and I think that will revive this approach as well.
Very exciting. It's a very exciting development that you described for us, Kim. Thank you very much. So here's a very interesting question, asking about what resistance means. What does it mean to be resistant? And are the mechanisms of resistance to antimicrobials similar across different strains of bacteria? And are they similar across fungi or other parasites as well? Or do different classes of pathogen have their own strategies to become resistant to the chemical treatments that we throw at them?
Yeah, I could take a stab at that. So the answer is both. So what we see, we appreciated this early on, is that there are plasmids. These are like mobile pieces of DNA that Kim mentioned. They're easily shared among different bacteria, and often they encode multiple different forms of antibiotic resistance, right? And so these can be spread across many distantly related bacteria. And so we could see the same resistance mechanism appearing in multiple unrelated bacteria, right? And this makes it very challenging to get on top of this and control this. Beyond that, there are specific forms of resistance. The gram-negative family of bacteria have that outer membrane that gives you this intrinsic barrier to the ability of many types of antibiotics from crossing the membrane. And these gram-negative bacteria have their own kind of efflux pumps that pump out compounds, although we see these as well in the other bacteria and gram-positives. There's there are ways of bacteria of mutating the target of compounds. We might see that these kinds of mutations might be tolerated in different ways in different species of bacteria. So there are so many mechanisms. Many are shared and many are specific. It's challenging to be able to really keep track of and catalog each of these so that we could really stay on top of resistance.
Indeed, it's a very challenging landscape. And so we're very thrilled that you both with your research groups are tackling some of this great complexity. Eddie, there's a question for you as to how targeted you can be in trying to do your screening for dark proteins that you think would have the best shot of then being therapeutic candidates. Do you want to comment on that one?
Yeah, absolutely. So this is, we definitely want to prioritize, be realistic in what could be the best targets that could be inhibited. And so, putting a target on the highest priority list is where it's located in the cell. If a target is located near the surface or on the surface, that certainly is the best target. And using bioinformatics predictions, we can predict where these proteins are in the cell. Using the fluorescence labeling, we can confirm where they are. And so certainly we're most interested in the ones that are most accessible potentially to inhibitors. So we're able to identify which those are. And we are pursuing some in addition to the one I showed you that are most towards the surface of the microbes.
Thank you so much. I think we have gotten through. Oh, there's one last one I'm going to throw out at you here. And then I think we've gotten through the wonderful set of questions. Thank you for asking them, everyone. Dr. Lewis and Geisinger, what is your take on bacteriophages as antimicrobials? Kim, would you like to tackle that one?
Yeah. So I've been following that field for a while. In the 30s, there was an American virologist who went to work in Georgia, and that is USSR, Georgia. And he introduced the phage therapy as they are to treat primarily infections caused by Staph aureus. With supposed success, they have never ran a clinical trial. It's a friend of mine who headed anti-infective division at Wyeth Ayers offered them money to run a clinical trial, and they refused to run a clinical trial. So that tells you a little bit about the success rate. In America and in general in the world, there's been a revival of phage therapy as a potential alternative approach. It has enormous promise because the phage attack bacteria, they multiply and they have the potential to indeed eradicate the infection. The downside is that the resistance develops rapidly to the phage and that there's immunogenicity. So you can, of course, inject the phage once, but what if you need repeated injections? The phage is covered with proteins and that may stimulate an exuberant immune reaction and death. But I am, I am cautiously optimistic about that approach. And I think that there are cases where it will probably work. There was a particularly dramatic one where a patient was infected with Acinetobacter baumannii, the DERI studies. It was multi-drug resistant. The patient was dying. And then there's a company that makes experimental phage for Acinetobacter baumannii, and that patient was given permission, his doctors were given permission to treat on compassionate basis with this completely untested approach, and he was cleared of infection. So we have this terrific precedent, which bodes well, I think, for that field.
Very interesting. Thank you so much. I want to thank both you, Professor Lewis and Professor Geisinger, for taking the time to share with all of us your insights today. I want to thank you for the outstanding work that you are doing here at Northeastern. We are enormously proud to have you on our faculty and to have you helping to lead the this huge aspect of human health challenge. We know that you have great ambition to take forward your own research and to build it even more at Northeastern to a really to an even more important aspect than it is right now. And I'm personally really supportive and excited to see where this particular field will go. But we have every optimism to share that the outstanding contributions you have made already will keep going and will multiply and will assist human health in the most powerful ways. So congratulations to you both on your outstanding work. Thank you for taking the time today to share with us, and thank you all who have attended. It's lovely to have you join us. It's lovely to have you be part of our Northeastern College of Science community, and we are very grateful to those of you who support us. Please do be in contact if there is any further connection that you need. Thanks so much and take care, everyone.
Thank you.